Hi, hello, namaste. A very warm welcome to bio classes. Previously, we focused on the raw materials required for that DNA construction, right? We observed major three raw materials, major the components of DNA. The first one is a nitrogen basis, second one is a pentose sugar, and third one is phosphate. In the previous class, we observed their structural formulas, how their structure we can write. So that thing we observed, and all the three materials together they will form a nucleotide. So in the beginning only I wrote there, nucleotide means these are the monomers of the DNA. That DNA is a polymer. So for the construction of that DNA polymer, the monomer molecule is nucleotide. How that nucleotide is formed? Nucleotide is formed by the three components: nitrogen base, pentose sugar, and phosphate. So today in this class, let us see how we can connect all these molecules to produce the nucleotide, and how the many nucleotides will form the DNA, and what are the speciality of DNA, right? So let me start with so first I will write the molecules. The first one, what is this molecule? Purine or pyrimidine? Yes, this is a purine, heterocyclic ring. And this kind of a molecule here, this is pyrimidine, right? So, and yesterday we mentioned the corners where nitrogen atoms are present here. In a purine ring, 1, 3, 7, 9. Keep this number in your mind. 1, 3, 7, 9. In these corners, we can see the nitrogen atoms. In case of a pyrimidine, we will see 1 and 3. So in the ring 1 and a third corner, we are having a nitrogen. So one is a purine, another one is pyrimidine. Okay. Next one, we are having a sugar. Which sugar? Pentose sugar. What is the name of the pentose sugar? Deoxyribose sugar. Why it is called as a deoxy? Why? Because in that sugar, from the second corner, that is from the second carbon, oxygen atom is removed. Deoxyribose sugar. So we used to write the structural formula like this, right? 1, 2, 3, 4, 5. 5 carbons, pentose sugar. So here at the second place, oxygen is removed. Okay, oxygen is removed. Now, here, here, and here. 1, 3, 5. So total how many carbons? 1, 2, 3, 4, 5. Total 5 carbons are present. At first carbon, third carbon, fifth carbon, we are having OH groups. Which groups? OH groups are present. Yesterday I had written that, yesterday I had drawn that diagram. OH group. And 1, 2, 3, 4. In these 4 corners at carbons, we are having hydrogen atoms. So this is the pentose sugar. Nitrogen bases, pentose sugar. What is the next one? Phosphate. So here, actually the phosphate molecule, I will write it as PO4 minus 3. This is the phosphate molecule. While combining, it is actually phosphoric acid. That phosphoric acid becomes phosphate. Okay. Now, including all of these raw materials, what we need to ready? What we need to produce? We need to produce the nucleotide. Okay. Now, in the case of a nucleotide, I will write here. Nitrogen base plus pentose sugar plus phosphate. Nitrogen base plus pentose sugar plus phosphate. All three together becomes a nucleotide. Okay. But so here nitrogen base plus pentose sugar, if only these two are combined means we will call this as nucleoside. What we will call it as nucleoside. Nucleoside, this much is nucleoside. When nucleoside combines to the phosphate, then the total structure is nucleotide. The total structure is nucleotide. That means which one is bigger? Nucleotide is bigger. Nucleotide, it is a combination of nitrogen base, pentose sugar, phosphate. In a nucleotide, if you remove the phosphate, then a nitrogen base, pentose sugar, only two, then it is nucleoside. Okay. Now we have to produce the nucleoside, then the nucleotide. So many nucleotides form on a polynucleotide strand. That polynucleotide strand is nothing but a DNA. Now, I am going to write some points here. Third carbon of pyrimidine nitrogen base combines with first carbon of sugar. This is one thing. Third carbon of pyrimidine nitrogen base combines with first carbon of sugar, or ninth carbon of purine nitrogen base combines with first carbon of sugar. Now, what it indicates? See here. Third carbon of pyrimidine. This is the pyrimidine, okay? Third carbon. This is the first carbon. First carbon, second carbon, third carbon, fourth carbon, fifth carbon, sixth carbon. Now, what is the statement here? Third carbon of pyrimidine nitrogen base. This is the pyrimidine nitrogen base. Third carbon combines with first carbon of sugar. So this is the sugar. This is the first carbon. This is second, third. This is fourth, fifth. First carbon of sugar molecule, third carbon of pyrimidine, both are joined. What happens? Both are joined. So now both are joined here. After joining, we are going to get nucleoside. What we are getting? Nucleoside. In case of this is the pyrimidine. Already we know. What are those pyrimidines? There are two pyrimidines. Yes, which are those? Pyrimidines. Yes, thymine and cytosine. Both are what? Pyrimidines. Suppose if it is a purine, if this combines with a sugar, then ninth carbon of purine base. This is the purine. So, which are those purine? Adenine and guanine. AG, adenine and guanine, both are purines. Ninth carbon, see here. Let's start from this carbon. 1, 2, 3, 4, 5, 6, 7, 8, 9. This ninth carbon of purine combines to first carbon of sugar. This is constant. First carbon of sugar. Then that is also forming nucleoside. What it is forming? Nucleoside. Just I told you. Nitrogen base combines with sugar. For combining, there should be one bond. That bond is glycosidic bond. So what is the bond name here? Glycosidic bond. So keep this thing in your mind. Yes. What is the bond name? Glycosidic bond. Glycosidic bond, it is found between the first carbon of sugar, third carbon or ninth carbon of pyrimidine or purine base pair to form what? Nucleoside. What is the connecting bond here? Sugar and nitrogen base connecting bond is glycosidic bond. And this glycosidic bond is formed by the elimination of H2O. So when these molecules are combined to the sugar, water molecule is eliminated and the bond is formed. That bond is glycosidic bond. So in that sense, suppose adenine, A stands for adenine. Adenine purine or pyrimidine? Yes, that is purine. Adenine combines with sugar. Here S means sugar. Adenine combines with the sugar. What is formed? Nucleoside. With which bond? Glycosidic bond. And that nucleoside name is adenosine. Sorry. Deoxyadenosine. So the nucleoside name is deoxyadenosine. Suppose if it is a thymine, thymine combines with the sugar. Thymine means pyrimidine. Thymine combines with the sugar. Then what is formed? Nucleoside. That nucleoside name is deoxythymidine. Deoxythymidine. Next one. Guanine combines with the sugar. Then its name is deoxyguanosine. Then fourth one. Cytosine combines with the sugar. Then its name is deoxycytidine. Okay. So what all these are? Four types of nucleosides. Four types of nucleoside. So here deoxy means here it stands for the sugar name. Adenine means nitrogen base. Adenosine means nucleoside. Suppose if I don't write a deoxy, if I just write adenosine, then this is also nucleoside. But it may be the nucleoside of DNA or RNA. But if I mention deoxy means it is the nucleoside of DNA only. So we are having four types of nucleoside. We got nucleoside. Now these nucleosides should combine with what? Phosphate. After combining with phosphate, then we will get nucleotides. Okay. Now so I mentioned here first one. Nitrogen base combined with the sugar. What we got? Nucleoside. Now sugar should combine to the phosphate. Sugar molecule combines with phosphate, not to the nitrogen base. Sugar molecule combines with phosphate. That means in between sugar and phosphate, again there is one bond. One more bond is formed. So now what I am mentioning here is sugar I am considering here. Third carbon or fifth carbon of sugar. Third carbon or fifth carbon of sugar combines with phosphate group. Combines to what? Phosphate group. There is no carbon, hence I have not mentioned. Third or fifth carbon of sugar combines to the phosphate. Now sugar molecule, come to here, sugar molecule. So already first carbon of this sugar combined to the nitrogen base. Now first corner will not work. Second corner also not working. Why? Because from the second carbon oxygen atom is removed, deoxyribose sugar. Hence it will not work. Now the working corners are third corner, fifth corner. Why? Because fourth corner combined to the fifth carbon. Now the working corners are third carbon and fifth carbon. Now third carbon or fifth carbon of sugar molecule combines with a phosphate group. Then what is formed? Nucleotide is formed. And here in between a bond is formed. So that bond name is phosphoester bond. What is the bond name? Phosphoester bond. And this phosphoester bond also formed by the elimination of water molecule. It is also formed by the elimination of water molecule. When third or fifth carbon having OH group, here OH groups are present. When third or fifth carbon combines to the phosphate, then the bond formed is a phosphoester bond with elimination of water molecule. Right? So this is the thing here. Now, so in that sense, if we consider deoxyadenosine, what is this one? Deoxyadenosine. Deoxyadenosine. So this is one nucleoside. Now the nucleoside combines to what? Phosphate. After combining to the phosphate, with which bond? Phosphoester bond. Then its name is adenylic acid. Or its name? Adenylic acid. Or deoxyadenylic acid. So you mention here. Deoxyadenylic acid. So in the same way, deoxyguanosine plus phosphate, then its name is deoxyguanilic acid. Deoxyguanilic acid. So if you mention the term acid last, then that is a nucleotide. If you mention the signs like this, then that is a nucleoside. Then deoxythymidine plus phosphate. Then the nucleotide name is deoxythymidinic acid. Then last one, deoxycytidine plus phosphate. Then the nucleotide name is deoxycytidinic acid. So what all these are? All of these are nucleotides. These are nucleosides. Nucleosides combined to the phosphate, we will get the nucleotides. Okay. So I'll mention here. All of these are nucleotides. Yes. These nucleotides are also called as nucleoside monophosphates. Yes, it's another name. Nucleotides, another name is nucleoside monophosphate. Mono means what? Single. Single phosphate. Nucleosides combined with the single phosphate means nucleoside monophosphate. Nucleoside monophosphates are nothing but nucleotides. So which are those nucleotide monophosphate? AMP. Adenosine monophosphate. Adenosine monophosphate. Then GMP. Guanosine monophosphate. Then TMP. Thymidine monophosphate. Then CMP. Cytidine monophosphate. All of these are nucleotides. Okay. Suppose here on best example. If this is combined with two phosphates again, AMP combined with the two phosphates, then what is its name? Adenosine monophosphate combined with the two extra phosphate. That means a three phosphate. What is its name? ATP. What is its name then? ATP. We have heard this term, ATP. Adenosine triphosphate. So that is the energy molecule. That means energy molecules are formed by the nucleotide molecules after the addition of phosphate. So that is the next another thing. So here, this is how we can see the nucleotides. So now, simply I am mentioning here how that a nucleotide will be looking like. So I am mentioning just this square means one nitrogen base. Any one nitrogen base, either purine or pyrimidine, whatever it is. Now, nitrogen base combines with what? 1 minute. This is one a nitrogen base. Nitrogen base combined to what? Sugar. Which sugar? Pentose sugar. Combines to what? Pentose sugar at first carbon. Right? Nitrogen base combined to the sugar at first carbon. 1, 2, 3, 4. This is fifth carbon. Now, nitrogen base, any one nitrogen base combined to the sugar, then it is a nucleoside. Now this is nucleoside. Now it has become nucleotide. That means it should combine with what? Phosphate. Now the phosphate combined to sugar at third carbon or fifth carbon. Now let us consider at a fifth carbon, phosphate is attached. P means here phosphate group. Now this is one a nucleotide. Nitrogen base, pentose sugar, phosphate. What is this one? Nucleotide. Now tell me, what is the bond present between here? Nitrogen base and pentose sugar. What is the bond? The bond is glycosidic bond. This is glycosidic bond. This is the first bond. Next, the bond present between sugar and phosphate. This bond is the bond present between sugar and phosphate. This bond is the phosphoester bond. What is the bond? Phosphoester bond. Now this is total together one nucleotide. Now nucleotide combines complementary with other nucleotides. Now the nucleotide combines with another nucleotide with a complementary. So here there is one rule. A combines with T, G combines with C. Adenine combines with thymine, guanine combines with cytosine. Okay, keep this thing in your mind. Now, I will consider this nitrogen base as adenine. What I will consider this one as? Adenine. This is adenine. Okay, adenine. Adenine combines with what? Thymine. It will make one pair with the thymine. So now the thymine is present in what form? Thymine is present in thymidilic acid. So I will write the same copy that side. This is a T, thymine. So I mention here, this is thymine. Okay. Now the thymine combined with the sugar. Here adenine combines to the sugar, that side thymine combines to the sugar. But orientation of sugar molecule is opposite. The orientation is opposite like this. Orientation is opposite. But this is first carbon here. Here also this is first carbon. But orientation is different. Now first carbon, second carbon, third carbon, fourth carbon. Now fifth carbon is present here. Now that fifth carbon combined to the phosphate. Okay. Now This is one nucleotide and this is one nucleotide. This nucleotide is adenylic acid, deoxyadenylic acid. This nucleotide is deoxythymidyllic acid. Both the nucleotides are joined. For joining, in between one bond is created. That bond is hydrogen bond. Which bonds are created? Hydrogen bonds. In between adenine and thymine. That means in between the base pair, in between the base, two bases, hydrogen bonds are formed. In between adenine and thymine, two hydrogen bonds are formed. So now this is one nucleotide. This is one nucleotide. Two nucleotides are joined like this. This is one nucleotide. This is one nucleotide. Two nucleotides are joined. Then it forms one base pair. This total is one base pair. Okay. In the same way, let me write another one. Another base pair, let me consider. So I will consider this one as a guanine. Guanine combines with, as usual, sugar, pentose sugar. Then here the fifth carbon, that is combined to what? Phosphate. Okay, same as it is. And guanine combines to what? Cytosine. Guanine combines to what? Cytosine. Right? Now, same as it is. Here it is combined to what? Sugar. Which is sugar? Pentose sugar. Okay. Then first carbon, second carbon, third carbon, fourth carbon, fifth carbon is outside. To the fifth carbon, here the phosphate is attached. So now this is another base pair. In between a guanine and a cytosine, there are three hydrogen bonds. How many hydrogen bonds? Three hydrogen bonds. Here two hydrogen bonds, here three hydrogen bonds. Total we are having three kinds of bonds. Hydrogen bond, glycosidic bond, phosphester bonds. We are having three types of bonds to connect all the molecules in the DNA. Okay. Now, this is one base pair. This is one base pair. Base pair should be connected. What should be connected here? Base pair should be connected. To connect the base pair, here what we can do? What is the carbon here? This is first carbon, second carbon, third carbon. Third carbon of this sugar make one bond with phosphate, downside phosphate, right? And this side, first carbon, second carbon, third carbon. Third carbon of this sugar makes one bond with phosphate, upper side on phosphate. Now, think here. This base pair, both are connected. What is the bond name here? Phosphoester bond. What is the bond here? Phosphoester bond. Now, we will call this together as a phosphodiester bond. Which bond? Phosphodiester bond. So, I'll mention here. So, phosphodiester bond. Right? Now, like that we can write all the base pairs. That's why I told you. Third carbon or fifth carbon of sugar. Third carbon or fifth carbon. Third carbon or fifth carbon. So like that, so many base pairs are present here. Like that. So third carbon or fifth carbon will combine. Now, see here. One interesting thing. This side and that side, sugar molecule orientation is opposite. Now here, fifth carbon combined to the phosphate. I will mention this end as a 5'. This side I will mention it as 5'. That side, this side here. 1, 2, 3. Here third third carbon, it is free. Here the third carbon is free, and that third carbon is having OH group. So there it is having OH group. Hence it can combine with another phosphate. So here third carbon is free. So hence I will mention this end as 3 dash or 3 prime. This is 5 prime, that side third prime. Now coming this side, first carbon, second carbon, third carbon. Here third carbon is free. I will mention this end as 3 dash or third prime. Coming to this side, first carbon, second carbon. Here second carbon will not work. Why? Because oxygen atom is removed. Last time only I told you. Third carbon, it is already had made on bond. Fourth carbon, fifth carbon. Here fifth carbon is outside. To the fifth carbon, phosphate is attached. Here third carbon is free. That means this side we will get 5 dash to 3 dash. If you write so many base pairs like this, means one side 5 dash, downside 3 dash, and that side 3 dash to 5 dash, 3 dash to 5 dash. That means we are having the DNA in this form. We are having the DNA two strands. This becomes one strand, this becomes one strand. So this strand 5 dash to 3 dash, this strand 3 dash to 5 dash. So we will consider this one as anti-parallel. Anti-parallel strands. Those anti means opposite. Anti means opposite. Parallel, both the strands are parallel, just like the railway track. How the railway track will be going, in that way the strands are parallel. ಸೊ ಎರಡು ಹಳಿ ಯಾವ ರೀತಿ ಹೋಗ್ತಿರ್ತೋ, ಎರಡು ಹಳಿ ಇನ್ ಬಿಟ್ವೀನ್ ಮಧ್ಯೆ ಕಾನ್ಸ್ಟೆಂಟ್ ಆಗಿರುತ್ತೆ ಡಿಸ್ಟೆನ್ಸ್ ರೈಲ್ವೆ ಎರಡು ಟ್ರ್ಯಾಕ್ ದಲ್ಲಿ. So like that the DNA both the strands are going parallelly, anti-parallel. Why? Because the directions are opposite here. This structure becomes DNA. So in the next class, let us consider the same structure, how it looks like a DNA. Why? Because the DNA means we are having this kind of imagination. This is our DNA. So the DNA should be looking like this, right? How this structure looks like that DNA. ಈ ಸ್ಟ್ರಕ್ಚರ್ ಆ DNA ತರ ಯಾವ ರೀತಿ ಕಾಣಿಸುತ್ತೆ ಅಂತ. Okay, so in the next class let us see that structure. Yes, thank you.